Hello, and we're going to go through the maths requirement of A-level biology, everyone's favourite. So I thought I'd just get the spec up and let's have a look through to see what the math skills are and see if there's any things that we need to know. So they'll have these codes, MS 0.1, etc. So students may be tested on the ability to convert between units, millimetres cubed to centimetres cubed, etc. Again, people struggle with this, just with like one dimensions 2 and 3. So for example, on this millimeter cubed to centimeter cubed, it's just a little tip is just to say that that 1 centimeter equals 10 millimeters. Write down that one first, and then just if I want to go to 1 centimeter cubed, then it's 10 millimeters cubed. Yeah, do 10 cubed, so that's 1,000 millimeters cubed. So write down the linear version, and then just cube it or square it depending on what you need to do is my little tip. Next one, I'm going to zoom through these, and it's basically if there's any that stand out, so you have no idea with that, then pop along to workshop or ask for a video on that one in particular. This is recognise and use expressions in decimal and standard form. Again, it's really common now that they ask questions in maths questions and they ask for the answer to be in standard form. So therefore, just be careful again. Again, it's more people who miss reading it in the question and therefore don't do it more than people who get it wrong. But we do get a lot of rounding errors. So if this was like 0.03 4 5 2 3, and I want it into let's say 2 significant figures, and I want it in standard form. So significant figures, as we'll get to in a bit, this 0 doesn't count, that 0 doesn't count. So these are the 2 significant figures. So a preceding 0 is not significant. So then this just becomes 3.5 times 10 to the, and I literally bounce my decimal point over. So 2 times 10 to the minus 2. Okay. A lot of people get the standard form right, but then haven't got the rounding right, so they'll put 3.4 times 10 to the minus 2. So be careful on standard form. But it's more reading questions for that one is the tip. What have we got next? What's the next page? Am I going across or? Right, next one. Use fractions, percentages, ratios. It's just reminding you that ratios need to be generally x to 1. We always make the last number 1. Even if I've got a female male ratio of I've got 2 females doing physics in a class where I've got 8 males. If it's asked me in a female to male ratio, I still make the male number equal 1, and therefore I need to divide the 2 by 8, and therefore that is a quarter, so it's 0.25 to 1. Hopefully I've got that right. It doesn't matter about matching decimal places on a ratio, but the number on the right hand side needs to be 1. Again, the tip is, if in a question, if it tells you it wants it in a ratio, write down like I've done here, in what order does it want it. So female to male, pop your raw data down, make the last number equal 1 and divide by the other one. I think the rest, fractions are straightforward, aren't they? Percentages. Estimating results is just, if it asks you to estimate, just estimate. Don't, you shouldn't really be getting your calculator out to estimate. MS 0.5, use calculators to find and use power exponentials and logarithmic functions. This is, the classic is the, trying to think of the example, like how many bacteria have I got? Well, it's 2 to the power of the number of cell divisions, because they double. the time to double. So if I've got 24 hours and they double every hour, well, it's going to be 2 to the power of 24. I've got 24 doublings, so that's to the power button. Really, the log is just making sure that you can use your calculator on that. Again, a lot of calculators have got that. It'll say log 10 button. Some of them just say log, and then they have a box instead, and you have to type in what you want the log of. So yeah, so log 10 is sometimes just written just as plain old log. They have asked about natural log in the past as well. So above the log button is normally, or to the side of it is the natural log button, LN, or log to base E. Again, I wouldn't worry about understanding what logs are and why you do it. I will just, it's just basically being able to use your calculator as it tells you what to do. Okay, so just be aware of it. And just be aware as well that on the log button, if you press shift, you get anti-log as well. So again, just be aware of that, because a lot of equations will have a log in, and they'll give you a novel equation you've never seen before, and you have to substitute numbers into it, as it talks about later on. So that's those done. Data handling. Yeah, use appropriate numbers of significant figures. So yeah, just look up the rules for significant figures. Again, I went through it before, which is the common one where people tend to be good, before a decimal place. That if it's 2, 3, 8, 7, 4, 2.9, and give the answer to two significant figures, people are generally good at putting well, that's 24, 0, 0, 0, 0. Yeah, as in it's 240,000. However, people tend to be worse at 0.0087325, putting that into two significant figures. That's just 0.0087. So just be careful on them. I keep saying just be careful because a lot of this is really basic maths, and therefore it's relatively straightforward. You just have to read questions carefully. This one about understand that calculated results can only be reported to limits of the least accurate measurement. If I've done 3 measurements of like heart rate, so I've done some press ups or something. I've got a heart rate of 92. I have a rest, then I do the press ups again, and it's 103, and then I do it again, and it's 100. Yeah, whatever the mean of those is. I haven't got a calculator, but let's say it's like it comes in at 98.7. I can't have a decimal place. Yeah, these are recorded to 0 dp. My mean therefore needs to be to 0 dp as well. Okay. So just follow the rules. If they give you a table of data and it's partially filled in, follow the decimal places as well. So finding arithmetic means. Yeah, again, add them up and divide by how many. Just be careful on your calculator. Make sure you're quick on your calculator as well. So that one's really straightforward, working out a mean. Construct and interpret frequency tables, diagrams, bar charts, and histograms. So this is all that it's construct and interpret. So remember, frequency tables tend to be like tally charts, or they'll have certain criteria of like height. So people who are I can't really go through all of this now, but I guess the important bit here is difference between bar charts and histograms. In a bar chart, the bars are the same width and there's a gap between them, and crucially, I'm labeling them underneath the bars. Okay, in a histogram, we have a linear scale like a line graph, and I've got my bars are touching. They don't have to be the same width because it might be frequency density, but crucially it's a continuous scale, and and yeah, this is frequency. Often, again, it can be frequency density. It's just scientists don't tend to use them because people don't understand frequency density. So, but yeah, just if you ask to draw them, make sure you've got gaps in your bar chart and not in your histogram. Again, that's interesting. Interpret data from a variety of graphs, e.g., explain electrocardiogram traces. Again, they can give you absolutely anything, and they will do. And just if you're given an unusual graph, and most of them are unusual, I literally run my finger over the x-axis so that I read it. I then run my hand over over finger over the y-axis and make sure I've read it. Maybe there's two y-axes, in which case if there's a blue line, I'll highlight the blue axis and the blue line with my highlighter pen that I've smuggled in, that I'm allowed anyway. And and my red line, I will do the same. So I make sure I know what's what, and then I just basically have my finger on the graph and I go up or with my pen. So as temperature increases, the rate of reaction is increasing. It's at a maximum rate at this point. So I kind of talk it through in my head so that I get it right. But yeah, novel data can be tricky. 1.4, understand simple probability. Remember, when we write our stats out, it's there is less than 5% probability that the difference is due to chance. You have to use those words in that order. So make sure you do that. Yeah, and we've been doing a lot of probability associated with genetic inheritance. So that should be reasonably okay. Understand the principle of sampling as applied to scientific data. Yeah, so you can be tested on your ability to analyze random data collected by inappropriate means, e.g. the Simpson index. So we've learned a version of this called as in our syllabus, there's a diversity index equation, loads of n's in it, and you have to know it. There are different versions of the Simpson index. Anyway, but they kind of do the same thing. They measure diversity. If they give you a novel equation, I think they gave that last year, like a novel version of, an equation we haven't met, but it was just putting in data into it. So that was straightforward. Again, there's if you have to know an equation, it has to be in the syllabus. Why is that not moving with it? Oh, it is now. 1.6, understand the term means, median, and mode. Yeah, again, it's important to know if mean, median, and mode are all equal, then the data is shows normal distribution, which means that you get that curve, like the bell-shaped curve, which is important for selection, directional selection, and understanding that. And they do ask you to work out the modes, so you do need to know mode is the most common. Median, you have to put them into rank order, or just cross off the smallest, cross off the highest. The median's really good for getting rid of outliers. If they're saying the average house price in the Rural Valley is this, they are giving the median average. They don't do the mean because the mean will be skewed by that £8.4 million mansion that I don't live in. 1.7, scattergrams to identify the correlations between two variables. Again, thing to watch out for there is just strength of correlation. So if there's a line on a scatter graph, it's not a line of best fit, it's a line showing the correlation. And if the data is close to the line, it's a strong correlation, so that's a strong positive correlation. This is a weak negative correlation because the points are more scattered about the line. And this is why we do stats instead, because saying it's strong or weak is a bit weak. Therefore, that's why we do the correlation coefficient or Spearman's rank to get an actual put a number onto our confidence. Orders of magnitude calculations is just image over actual. Again, it's knowing your units as well, micro, nano, etc. Again, people tend to be good on them on length, but it's just as soon as you change to volume, people struggle. 1.9 is stats test, so hopefully you're okay on these. Again, none of them they will always give you the equations for them. Look how it says correlation coefficient. It doesn't even say Spearman's rank. So they're unlikely to give you a correlation coefficient to do, because different centres do different versions of that stats test, and therefore it will be an advantage if yours came up. So yeah, the one that they generally ask about is chi-squared, because it's also in the spec for genetics. However, they could ask about t-test, but they would have to give you the equation. Yeah, it does. Yeah, students may be tested on their ability to select and use. So you need to know when to use the stats test. So I always think student t-test, I do that first. That's when I've got two means, got mean values. Correlation is when I could draw a line graph. Yeah. So I've got a range, and chi-squared's all about individuals, or it's not t-test or correlation. Next, understand measures of dispersion. Yeah, standard deviation and range. Really, it's knowing the advantage of standard deviation over range. I always give the example of, that if I was in the classroom, the range of ages would be I would skew the range really high up. However, I would have less effect on the standard deviation. Standard deviation, all the data contributes to it, whereas range, only the low, like min and max contribute to it. So there's only 2 bits of data there. All the data goes, and therefore it's less skewed by outliers. As it says here, you've got outlier results. Again, you wouldn't call it an anomaly, it's just an outlier. What we're doing here now, uncertainties. Again, you've done an exercise on that. I haven't time to go through all of that now, but you do need to know how to combine uncertainties. But yeah, I can do a video on that if required. Then we get to algebra. So yeah, the less than sign, the very less than, or a lot less than, a lot greater than. Yeah, do we know this one? Yeah, that's proportional to. Proportional to. You meet that in Fick's law, where the rate of diffusion is proportional to the surface area times by the concentration difference divided by the thickness of barrier or membrane thickness. Yeah, and this one's approximately. Change the subjects of an equation. Again, they tend to give you like a weird equation, and you have to change the subject of it. So just remember, whatever you do to one side, you do to the other. Yeah, so magnification seems a bit easy, but if it was y equals mx plus c, I don't want it in terms of x. Right, well, I need to minus c from both sides, so y minus c equals mx. Right, I need to divide both sides by m, so x equals y minus c over m, etc. Just do it in steps, like you were taught. 2.3 is just about putting numbers into equations. Again, just do it carefully. Generally straightforward. But yeah, brackets can cause a problem. So when you square and when you divide, just be careful. Solve algebraic equations. Yeah, so they will give you, codec output equals, often they give you the stroke volume, or that's from a graph, and you have to work out the heart rate from a graph. So you have to work out heart rate and stroke volume to then come up with the codec output. That shouldn't be that tricky. Logarithms in relation to quantities over several orders of magnitude. So yeah, so it's just being aware of a log scale. We've done a bit of log scale work this year. Remember, you just need to know how to use and not really worry about how to understand it. But it just means that you can plot a massive range of numbers. Yeah, so if I'm trying to plot 10, 20, and 5 million, I would never fit that on an axis. Therefore, by using a log scale, I can fit it onto an axis. And then graphs, which I think I feel like I've talked about graph before, but it appears again. Yeah, this is just translate information between graphical, numerical, and algebraic forms. Again, they do lots of this, presenting data as graphs and just understanding it. Plot two variables from experimental. Again, this is repeating from before, but yeah, you need to be able to plot all these graphs. You've done loads and loads of line graphs, but they have asked bar charts in the past. Histograms, they've asked about graphs and scatter graphs. Again, I've never seen a scatter graph being asked, but you never know. You need to know y equals mx plus c. Remember, m is the gradient, c is the y-intercept. Sketch the shape of a graph with a linear relationship. Yeah. Often there'll be one line on a graph, and they'll ask you to put on, what would it look like at a high temperature. Determine the intercept of a graph, e.g., finding the compensation point. Yeah, so if you've got some data, I think the classic one is, I've got some photosynthesis data that looks like this, but I can extrapolate back, and I can find the compensation point, that is the concentration, the light intensity that causes no bubbles. This is like number of bubbles. But yeah, look up compensation point. That's where it's the x-axis intercept, and then you can do the y-axis intercept for the amount of respiration that's going on. Rate is just like delta. Still can't do a delta. Some space. Delta y over delta x. The change in y over change in x. And if it's a straight line, it's nice and easy. I just make it into a triangle. So that's on this one here. If it's a curvy line, then you have to draw a tangent at that point, and again make a triangle. So there's my graph. There's my line. I want to know the initial rate. Right. Well, there it is. Make it nice and long, your tangent, and then just get your y and x difference. Just make sure that you get the difference and not the actual scale, because the scale might not start at 0, it might start at 30, go up to 50, and it's gone along 10. Therefore, it's 20 divided by 10, and then whatever the y-axis value is, that becomes the first bit of the unit, and whatever the x-axis, which is going to be time, becomes the second bit of the unit. And last one is well, it can't be. It is. I've got through all of them. Wow. So this is calculate the circumference, surface area, volumes of regular shapes. Again, they tend to, if they want you to do a sphere, they'll give you the form of the sphere. A cylinder, you probably need to know, and this is that one's part of like using a potometer or a respirometer. That there'll be a capillary tube. So there's a little fine tube in a glass tube, but that's got a round cross section, and in there's like a bubble moving along, and as it moves, it draws out, as in it describes a cylinder. So if it moves to there, it's moved distance L from the bubble there to the bubble here, and therefore the cross-sectional area is pi r squared multiplied by L gives me the volume of liquid that's moved along that capillary tube. So a lot of people, when asked for that, it'll say, give your answer in millimeters cubed per hour, and they've run this experiment for 20 minutes, and they've got like a length of it's traveled 5 centimeters in 20 minutes. Right. Well, I need to get it into millimeters, so that's 50 millimeters. Get the units changed first of all. Then I need to do 50 times pi r squared, because that's just the distance moved, not the volume. So that'll throw out a certain volume. So it's like 160-ish, approximately 160 millimeters cubed. But that's per 20 minutes, per 20 mins. Therefore, I need to times it by 3. So is that 480 millimeters cubed per hour. Yeah, and in terms of like unit conversions, which hasn't really appeared in these math skills, but it does appear reasonably often. Is yeah, just take the units one at a time. If you need to convert, again, the classic one, if we're thinking like respiration, it's going to be something about like the amount the capillary tube moves. It's moved like oops, where's the pen? 20 millimeters cubed per hour per it's going to be like per kilogram body weight. Yeah. So if we pretend that those are the units, and I want to get it into, oh no, I want it into micrometers cubed per minute per gram. So yeah, just take them one at a time. The hard one is the volume. Oops. So remember what we did before. So yeah, so 20 millimeters equals, well, how many micrometers is it going to be? Well, yeah, sorry, the 1 millimeter equals 1,000 micrometers. So 20 millimeters is going to be 20,000 micrometers. So 20 millimeters cubed equals 20,000 micrometers cubed. Again, it's going to be give you a massive number. So that's a hard one. Going per minute, well, going from per hour to per minute, I just think it through, thinking, well, there'll be less per minute than per hour, right? And I know there's 60 minutes in an hour, therefore, right, I need to divide by 60 to get it into per minute. Yeah, there's going to be less per minute than per hour. And the same for per gram. Well, a gram, if I had a gram of biological material, that's obviously going to be doing less respiration than a kilogram of biological material. So yeah, so I need to divide by 1,000 as well. So those I just think through logically. They tend to be an easier conversion, but yeah, they do crop up quite often, do unit conversions. Right, I've been on going for like 28 minutes there, so I better stop. Hopefully amongst there, I did this something useful.